going to be presented from Regensburg by Gerhard Liebisch, titled Quantitative High-Throughput Lipidomics, Methods, Applications and Pitfalls. My name is Valerie O'Donnell. I'm based at Cardiff University in the UK. As usual, the webinar is being recorded. It'll be uploaded onto the Lipid Maps website and YouTube channel in the next 48 hours. We've got 45 minutes for talk and then about 10 minutes at the end for questions. And how this works is that you, as the talk is going on, please post your questions as you think of them in either the Q&A or the chat, it doesn't matter which. Because what that does is that at the end of the talk, it allows me to unmute you and let you ask your question yourself. So if you get your question in early, you'll be guaranteed being allowed to ask it. So post them as we go along, don't save them till the end, just keep posting. We'll take as many as we can. So today's webinar is being sponsored by Avanti. We're grateful for their ongoing support. And now I'm going to introduce Gerhard. So Gerhard Liebisch was chemistry undergraduate and also obtained his PhD at the University of Regensburg. He did his PhD in sphingolipid metabolism in Tangier disease with Gerhard Schmitz. He's currently team lead and research associate at the Institute of Clinical Chemistry and Laboratory Medicine at the University Hospital of Regensburg, being responsible for instrumental analytics lab. His research interests focus on the development of mass spectrometry methods for quantification of lipid species, a hugely important area, particularly for all the rest of us who focus more on the application of these methods for biological questions. I mean, the work that Gerhard and his colleagues does in this area is absolutely fantastic for the research field, so really important. And for more than 20 years, methods developed by Gerhard and colleagues have been applied in large-scale clinical studies and basic research, including the use of stable isotope-labeled lipids, to trace transport and metabolism of lipids. Gerhard is co-author of more than 200 papers in peer-reviewed international journals and editorial board member of Journal of Lipid Research. Some of his most cited work, with citations around 500 to 1,000 each, are one, I think, which exemplifies the method approach, lipid extraction by methyl terbutyl ether for high-throughput lipidomics, which is a really useful and really important method that is widely adopted nowadays for cohort work. He also published on ABCG1, human homologue of Drosophila as a regulator of macrophage cholesterol and phospholipid transport, and molecular cloning of human ATP binding cassette transporter for sterol-dependent regulation in macrophages. Gerhard is passionate. I think one thing that I think about Gerhard is that he's passionate about upholding rigorous standards in lipidomics analysis, and this is really important. And in this regard, he's a co-founder of the LSI, Lipidomic Standards Initiative, and a board member of International Lipidomics Society. He's one of those people that I know if I have a question about methods, and I'm not sure if I'm doing something right, he's one of the few that I will go to and I know that I'm going to get the right answer, and a thoughtful, useful answer. So thank you, Gerhard, for that, for ongoing help and support. A couple of the things I think to flag, recent initiatives in that regard that Gerhard has pioneered, led on and co-led. One is the shorthand nomenclature for lipidomics annotation, realizing a number of years ago that there were problems when, the kind of untargeted MS only methods came out. Annotation was being done in a way where people were over annotating structures horrendously and giving, really names right down to chirality, for example, when you only have a mass value in lipidomics is really poor practice. And Gerhard led with others, including Fritz Benner, Harold Kufeler and a number of others, on developing a shorthand nomenclature, which has been adopted hugely across the field, and we now have embedded in Lipid Maps with a dedicated search interface and helpful information to support this. Another initiative which has been really on the cards for the last few years is around standardization of methods. Together with colleagues in Lipidomics Standards Initiative, there were two papers I think worth flagging. In 2019, Lipidomics Needs More Standardization, which was a statement paper by the LSI. And then more recently, the Introducing the Lipidomics Minimal Reporting Checklist, Bowden, McDonald, Cruz, Gerhard, Ahrens, and many others, really trying to get lipidomics to the sort of standards that other fields have adopted, such as for example, data deposition in transcriptomics and this kind of thing, where we really need to be thinking about how we're doing experiments and then reporting in detail exactly what we did so that others can either repeat or check or whatever. So I think this is my introduction for Gerhard. I'm delighted to welcome him today and look forward to hearing your talk.
Yeah, thanks a lot, Valerie, for this kind introduction. And yeah, that helps a lot because you mentioned already several points I would like to discuss on the next slides with you. You will see preparing this talk, and Valerie pointed that also out, our main focus is methods and I will also talk more about pitfalls, applications, some, but not so many probably. And of course, I will also discuss or mention the recent things we initiated concerning standards and also the checklist. So let me start with one of these slides. I love really to reason why we should quantify lipids. I think if you look at that picture, you immediately realize that it's important to get really numbers on these hundreds of different lipids which are present in a simple cell membrane, because without the numbers, which means the moles, we cannot not get the function. The biophysical properties are related to the concentrations and the composition of these molecules, and therefore I think that is just an easy example why it makes a lot of sense to quantify. I will give you more of that because I think it's key to get more insight and also comparability, and also for the standards, quantification is a very important factor. So at the beginning, I would like to introduce our concepts, our setup, and show you how we see quantitation and how we use the different methods. So, you know, lipidomic workflows, they start with lipid extractions, and also pre-analytics is a very important factor to really get the accurate and the real native lipidome, I would say, because there may happen a lot. We learned also a lot in the recent studies we did in our lab, because we thought that is an easy step, but it is not. I hope I could convince you. There are two main workflows. One is direct infusion mass spectrometry, the other separation. And we use actually both of them. And you can couple to low resolution, usually triple quad instruments, or also high resolution instruments, the more fancy instruments, which has some advantages. I will also show you. And the next step is how we identify lipid species or molecular lipid species, and at the end, quantitation. This is a last step, but also a very important one. And here in orange, some factors which are worth to really evaluate in your method development. This is stability, recovery, also some interferences. This may be isobaric or isomeric in nature, and the lipidome has a lot of overlapping things in source fragmentation. And at the end, we try really to characterize our methods concerning reproducibility, accuracy, limit of detection, the linear range, and also sample carryover. And I think that is also what we ask in the checklists. And this is really, if you have a really good validated method, that it's easy to do studies and also to compare data with other labs. So I think that is worth to do that work. So what is our aim in method development? Quantitative, high throughput, because we are a clinical lab and we need really reasonable time for each sample. So the methods should have really a short running time. And we do a lot here with flow injection analysis. Initially coupled here to tandem mass spectrometry, but now we use more also high resolution mass spectrometry for that. And of course, you cannot do all the stuff with the direct, but you need also here LC coupled methods. And you can analyze a lot of different materials. And in particular, I like applications there when you introduce stable isotopes, because then you can really analyze the transport and metabolism of these lipids. And this is much more functional to my opinion. I will show you also a bit on that later. So this is the basic setup we are using, and it started all out here with a micromass machine, more than 20 years ago. And this is a triple quad instrument, and for the flow injections, and we can really cover here the main lipid classes, phospholipids, sphingolipids. For the free cholesterol, there is a derivatization method, the acetyl with acetyl chloride. And recently, and Marcus in our lab did most of that work. We also use here the Q Executive for these direct methods. And there we have the beauty, the triglycerides. I think this is very nicely you could do with the flow injection with the high resolution. A direct way of getting really the comprehensive picture of that. And also for the free cholesterol, you don't need derivatization. And I told you we use also LC. When we use LC, this is for the minor, glycerosphingolipids and also sphingolipids. Here I will show you here sphingosine 1-phosphate APA example, or the minor sphingolipid classes we have for methods, and also for the bile acids. If you have isomeric lipid species, you need separation, and then LC is frequently a must, because you will not get specific fragmentation of some of these molecules. So the bad thing is, our old instrument really quit service a couple of months ago. But the good thing is we got the replacement. Not ready in operation, but same principle. It's now a Waters instrument, and we hope we can get some improvement in terms of sensitivity and also speed with that instrument. And these flow injection methods have the beauty that you need only a few minutes. For instance, here we have a lipid class specific approach, and there is 1 minute per lipid class per sample and here we have about 5 to 10 minutes per sample with a high resolution. So you see, the speed is quite sufficient, with these flow injection methods. And with the LC methods, we aim run times with a few minutes. Let's say 3, 4, 5 minutes run time. This is acceptable for our purpose to have, let's say here around 20 samples or 10 to 20 samples at least per hour. And then you can do really reasonable studies. And here the good news is also we got here also the brand new machine, or not the very latest, but a significant improvement in sensitivity. And this helps a lot for them of these minor species. But also here we need to transfer the methods and really to evaluate sensitivity and then also linearity and these things. So shotgun workflow. This is an easy sample preparation. It's also advantage. Here we have a roboted lipid extraction. We have a pipetting robot which recovers here chloroform. Where we mentioned this MTBE method. That was more Dominik Schwutke's work, but we did comparison also, but it works also nicely. But due to historic developments, we still use the Blantyre as our main method, and the robot recovers the chloroform phase, and there we infuse simply these crude extracts. And how do we quantify with these methods? And it's all electrospray, and you all know in electrospray we have this matrix interference, which means the ionization depends on what these spray droplets have as components, or which buffers, which composition of solvents you have here in using this, and this makes it difficult or to quantify without an internal standard. So the ideal situation is to have your analyte and your internal standard in the same droplet. Then you are safe. Then matrix effects are easy to compensate usually. And this is, you can imagine, quite easy if you have a flow injection method, because then you infuse your internal standards with your analytes together. And this is how it looks like for, here one of the major lipid classes, the PC, phosphatidylcholine and sphingomyelin, and you all know both have a phosphocholine head group, and if you do it in positive ion mode, your fragmentation, you get a fragment of 184. And this is a precursor ion scan, and you see all of these different species. And if you do want to do quantification, you add internal standards. We use here the 28.0 and 44.0. And we do a kind of matrix calibration, which means if we analyze plasma samples, we have a plasma pool and spike here different species on top of the plasma pool we extract, and so we get here calibration lines for different species which have different chain length, double bonds, and so we get different responses, and that can be used to calculate all the different species. And you see also if you have really different responses for all the different species in your system. So I can recommend to check how is the response in your system of different lipid species, because that gives you an impression, do I need a correction, or is it fine just to calculate the ratio of my analyte to internal standard? And this is for this lipid classes, it's more or less fine. You see not big differences depending on the chain length or the number of double bonds. They ionize very well, and also fragmentation is nice in positive ion mode. So that is an easy example, but I will show you later an example where correction and response corrections may be necessary. So what is difficult, let me go back a bit. You see sphingomyelin and PC species, and of course you see here some overlap, especially if you use here low resolution mass spectrometry, you have this type 2 overlap, And here is a picture which depicts this type of overlap. This is present more or less in all the lipid classes which have fatty acyl chains, because if you have here one double bond less, you have a second isotopic peak which overlaps here the monoisotopic peak of a species which has one double bond less. Here PC36.1 overlaps here PC36.0, of course. And this is an isobaric overlap, which means this is not exactly the same mass, Of course, these are not isomeric, these peaks, but you have a small mass difference, about 9 millidalton. So no chance with a low resolution mass spec to resolve it. With a high mass resolution, you may have the chance, but you need to check. You need to have a good instrument. Show you later some details on that. But if you have no resolution on that, you need to do some modifications, some deconvolution, and that is an example. We checked an algorithm. We spiked here, the blue-colored species on top of a plasma in increasing concentrations. Here you see that sphingomyelin species. You see a nice rise, with the increasing concentrations, but you see also a rise in the PC species, which has one Dalton, which has a 1 Dalton higher M/Z ratio, And you can imagine, you have also in without spiking, of course, this natural overlap, and you need to correct. And you can do this just based on the relative isotope abundance, which you can calculate. And if you do that, you see, this remains constant. And in particular, in this precursor ion scan, you have a huge overlap. You see, if you do not correct, for instance, for that sphingomyelin, far too high values. With a correction, due to that overlap, you see it's only a minor species. So that is obviously necessary. to address this type 1 effect, and it's also necessary to check it in high resolution mass spectrometry. And Marcus did that for our Q executive, and here we have only mass resolution of 140 K at 200 MZ. And Marcus did a nice experiment. He uses here a constant amount of here LPC 18.0 and did an increased spike of the more unsaturated species. And then you see in that case, you may resolve here this +2 isobaric peak of the lyso-PC, but yeah, not so nicely, not baseline resolved, even at this highest resolution with our instrument here, cholesteroliste. Yeah, you see, you get some mass resolution, some resolution of these peaks. But if you go to higher masses here for the triglycerides, you see more or less one peak. Yeah, so mass resolution decreases in these instruments. So he evaluated that in detail because these near isobaric peaks in this orbit trap instruments may lead to interference. Yeah, you see this the black colored peaks. This is a simulation of that overlap, and in red you see this is what we measured. Yeah. And you see, this is decreased. Yeah, these signals are decreased when you have this overlap. Yeah, in some cases, and Marcus quantified here, and it may be of advantage if you use, when you have this overlap here for the cholesterol ester 18.1 to 18.0. If you use the first isotopic peak, yeah, these are these squared values. There you see you are closer to the target value. This is a dashed line, shows the target value. And I just want to mention, if you do here correction, initially we thought, for instance, for the triglyceride, we need to correct like we do for the triple quad. You overcorrect actually. Yeah, you get here in that case, of course, you have some resolution, but you would really get negative values. So that needs to be checked for your particular system, and your really your depending on the mass resolution of your system. So there may be other isobaric overlaps, and for instance, from sodiated ions. Here is an example of the PC 36.1. Usually we use here the protonated ions. But for instance, if you run a helix separation, you always see also a bit of sodiated peaks. And this have here 22 Dalton higher mass. And this is isobaric here with the PC 38.4. Yeah, this is 2 carbons, 3 double bonds. And here you have only a small mass difference, yeah, which is only 2 millidalton. So this even good orbit traps frequently cannot resolve. Yeah, this is very hard to resolve. So Marcus checked whether we can do here also some isotope corrections, and you can do. You can calculate based on the internal standards. Yeah, you have here, for instance, the PC 28.0, which has actually here this sodiated ion 36.3. So you can calculate the ratio of protonated to sodiated and use that ratio to correct in positive ion mode this overlap. Yeah. And here he did it for a plasma study with more than 200 samples. And if you do not correct here for the PC 38.5, which gets a huge overlap here from the PC 36.2, you really overestimate, yeah, your concentrations. If you correct, you see these the blue dots. You get a very nice correlation of the negative ion mode concentrations, which we use normally, with these corrected values. Yeah, This is more or less identical, but sometimes it's advantage to analyze, for instance, PC in positive ion mode. Then this such algorithm may be advantages. So species response, I already mentioned that also that needs to be considered if it's hugely different. And this is the case if you analyze your cholesterol ester. Non-polar lipid species, they are ionized frequently as ammonium adducts, and they give nice signals. But they have really huge differences in response. And here you see also that the more unsaturated species, they show higher response. And Marcus quantified these responses, and you see there is also some dependence on the number of carbons. Yeah, you see higher response with a higher carbon number, but the most pronounced effect is the number of double bonds. And why these cholesterol ester show higher responses? Because you have here one effect, and this is in source fragmentation of these ammoniated ions. They lose here the acyl chain. And this is related here to the number of double bonds. So the more unsaturated these species are, the more stable these ions are. And that could be at the end put into a model. Yeah, and you could calculate the corrected values based on that model that contains your carbon number, double bond, and additional factors. And if you do so, you have a nice correlation. This is here the dark triangles. This show nice correlation of enzymatically and FTMS determined concentrations for plasma samples. And if you don't do, with the uncorrected, you see an overestimation, yeah, which is basically based on this higher response of the unsaturated species. So here, and I would like to say that also for other non-polar lipid classes, triglycerides, you may observe similar effects, but there it's even more complicated to do the species response correction. So we don't do it, though it's more for the accurate quantification. Comparison is, of course, still possible without these response factors. But if you want to really have accurate quantification, you need some corrections. So co-elution of internal standards in LC methods. Yeah, there is often the discussion: does it matter or doesn't it? Let me show you that cartoon. You have two different modes in lipidomics analysis. One is polar selectivity in the helic methods, or if you use helic also with the SFC, the polar separations, you get head group the polar selectivity. So one the lipid classes have more or less co-elution, and this is frequently also co-elution with your internal standard. And if you use reversed phase chromatography, you have a non-polar selectivity. You separate the different acyl chains. And if you think of quantification, as I told you, internal standards should be ideally in the same droplet. Then this situation is, of course, better than that situation. Yeah, and therefore we are great fans of helic chromatography. This is a quite old example. Yeah, there we did lysophosphatidic acid and sphingosine 1-phosphate, and here you see nice co-elution with the internal standard 17.0 of the natural occurring LPAs, and also for the sphingosine 1-phosphate. And to give you some data, how you can really improve with a good internal standard. Sabrina in our lab did that for the bile acids. This is just acetonitrile precipitation. We have stable isotope labeled internal standards for each of the species, but at that time, not for the tauroconjugates. This results in not so nice accuracy and precision. So, but they became available. Nowadays, you can buy most of these labeled compounds. And Sabrina did simply calculation, she calculated here with the matching internal standard for the tauroquinodeoxycholic acid. Here we have a D5. This is a newly available standard at that time. And you see, if these are the QCs in our clinical routine assay, this is really an improvement compared to that. We used before here the D4, the glycoconjugate, which has a bit different retention time compared to the tauroconjugate in our method. But it's a different conjugation. So you see, there are some outliers. So clearly improvement for the quality controls. And she did the same for the patients, and you can easily see that if you use a different tauroconjugate, which has a different retention time for calculation, you are off the target value. Same for the quality controls and also for the patient samples. And it is the last, the red colored peaks and also values here are calculations just based on counts. And here you see you get much higher variation compared to the proper internal standard, and you may have here twofold changes because you get not accurate compensation of your effects, matrix effects and other effects. So a good internal standard, I can really say it's key to get really accurate quantification, and co-elution improve that. A clear message, and I think it's easily explainable if you think of this ionization process. So now to the pre-analytics and extraction. I told you, we thought extraction, this is easy going. We have here the Bleiuntire and we just extract, but on the next slides, I would like to show you it is not. But first, some words on pre-analytics. Especially if you go for lysolipids or other unstable products, it could happen so much things here for the lysophosphatidic acid or sphingosine 1-phosphate. If you leave it at room temperature, the whole blood, or even here the separated plasma, you see a huge increase. Separated plasma only for the LPA, but in whole blood for both, sphingosine 1-phosphate and LPA, there is rises several fold. So there is only you need to really separate fast and freeze samples if you want to preserve the real blood concentrations. Also for tissue, we thought, extraction is easy. But we learned a lot during that two studies here. Sabrina and Alex investigated different tissues concerning the stability of homogenates. And there was a second study, Marcus was spearheading about homogenization and homogenization of solvents in liver. And I just would like to mention here we used usually in our lab methanol water for homogenization of these tissues, because we think there is not a perfect precipitation and we can easily handle the samples. But our thought was this huge fraction of methanol will really stop enzymatic processes. But I really can say no, it doesn't. There is happening a lot, and I don't want to go to this whole details of the publication, But if you check, for instance, ceramide sphingomyelin ratios, you really see a huge rise within a few minutes, and also during freeze thawing, you see really a huge rise in ceramide, and also in other lyso species. So there is quite a lot of lipolytic activity in these samples, despite you have here 50% methanol in your sample. So really, yeah, this needs care. And in that study, Marcus, I have not the data with me, we learned that homogenization is really key, and this it's not sufficient if you grind your sample and put solvent to that. It's really, we use always now a bead-based step in addition, to have really a homogeneous, dispersed sample to get proper lipid extraction. And instead of these tissue extractions, I want to discuss with you here lipid extractions of plasma. And we checked the extraction performance of one-phase lipid extractions, because they get much more popular, and I think they have clearly advantages. Yeah, you just put your organic solvent to the plasma, mix it, and then centrifuge and take the supernatant for your analysis. And we wanted to evaluate the performance, and therefore checked how much of the lipid is not found in the supernatant, but in the pellet. Therefore, we did an additional step here. We used the Bly-on dia, yeah, this chloroform-based extraction, and re-extracted, yeah, supernatant and pellet. And for the pellet, we did bead beating, yeah, to have a proper extraction. Of course, we need to check our own method. This is the extraction recovery of the Bly-on dia, and you see for most lipid classes it performs very well. Yeah, you may lose here some lysolipids, yeah, because they are a bit too polar to go to the chloroform phase, but still this is in a good acceptable range. This was done by flow injection with the Orbitrap. And these are the data. I want to focus your attention here first on the pellet fraction. Yeah, this is a red line, and we sorted the lipid classes in increasing polarity. Yeah, and we also sorted the solvents in increasing polarity. So on the left, you see Bly-on dia as the reference. Here we have the target value, if you want. And if you look at the red line for the lysolipids, yeah, you lose only minor portion. This is very good. But if you have a look at the PC as a standard phospholipid, if you here see the acetonitrile, you lose already a substantial fraction of your PC in this pellet fraction, yeah. And this gets even worse if you go to the non-polar fractions. And there you see also for the ethanol, for instance, yeah, the cholesterol ester you almost completely lose in the pellet fraction, yeah. And this is, I have to say, sample to solvent ratio 1:3. And we checked here in the next slide here whether solvent, the increase of the solvent ratio improves the situation. And there we did it only for BUME, butanol methanol mixture 1:1. It's isopropanol methanol, and here 1:3, 4, 5. And you can easily see if you use a higher solvent ratio, you can improve the situation. Yeah, you lose less of the lipids here for the sphingomyelin in the pellet. Yeah, for the methanol, you easily see you can improve the solvent fraction. For these non-polar lipid classes like the TGs or cholesterol ester, you will still lose everything more or less. Then you may ask, how could I change the situation, or compensate this loss if I add here an internal standard? Yeah, and we did this, therefore, the same experiment with the equi-splash, which was added here to the samples, and checked where can we found the equi-splash, which is from Avanti, this deuterated mixture. And you see that behaves similar, yeah, in most cases, but there are some differences. Yeah, if you look here for the TGs, the endogenous TGs were show a better recovery here in green in the supernatant than here the equi-splash, because we did it like that. We put the equi-splash to the cup and removed the solvent. And but I have to say we really used sonication and also to really recover this standard. And finally, we to get the complete picture, we added the internal standard with the extraction and did that with for the high, intermediate, and low triglyceride samples. And you can immediately see for most of the lipid classes, one phase performs well. But if you go here to the triglycerides, cholesterol esters, yeah, you see some strange results. We can explain these results, but this is simply because these solvents do not dissolve these non-polar lipid classes. Yeah, and this is a big issue. You should check in if you use this kind of lipid extractions. Yeah, especially these polar lipid, non-polar lipid classes may not recover nicely. So now to the applications. There are only a few minutes left, but just to mention, of course, high throughput method can be used for large studies. We did that here for a cohort with almost 4,000 samples, and this was cardiovascular risk factors. And yeah, same result as different other groups found it. Yeah, as you see, some risk for ceramide species, but not some are with a higher risk. Yeah, the 24-1 and the 24-0, for instance, has a lower risk constellation for cardiovascular death. And yeah, similarly, you could use plasma lipidomics to really do genome-wide association studies. Yeah, this was done here in a 5,000 cohort some years ago, and there were nice correlations as a proof of concept also with the enzymes. Yeah, this fits nicely. And yeah, that study, I think Michael Hoczabek presented already during this series, and I have to mention this is all quantitative lipidomics, and for pancreatic cancer, and this is a nice example that you get comparable results in different labs. And I want to discuss more that technique. This is stable isotope labeling of glycerophospholipids. And this is quite straightforward. You can feed cells or mice. I will show you some data, some spectra from mice with these stable isotopes. Here, D4 labeled ethanolamine. So usually we analyze here the PE with the neutral loss 141. And if you feed here this deuterated one, you get the 145. Yeah. Similarly, you could do this for the D9 choline. And if you check the pathway in the liver, you have a conversion of PE via methylation, yeah, of this amino group, and though you can convert PE to PC. And if you have a D4 labeled PE, you get a D4 labeled PC. And so you may nicely differentiate the PCs coming from the PE with the precursor ion scan of 188. And the choline derived species, you get here 193. And this is mouse data. Yeah, you can here IP these labels into the mouse. This is 4 after our IP, and you see a nice spectra for the labeled PE species, which more or less similar that than those species already present, the unlabeled. For the PC, it's more interesting because you see that the PC species derived from choline incorporation, the Kennedy pathway, are a lot different from that derived from the PE methylation. Yeah, here you see much more unsaturated, which is well-known fact, but that is a nice example, and I think, yeah, worth to use these kind of approaches. The final example is here a recent cancer study, colorectal cancer, together with Josef and in Munich, and also Klaus-Peter. And there we studied 2 cohorts in our labs. Yeah? This is colorectal tumor tissue, and found here a higher fraction of these triglycerides. Yeah? This may be of dietary origin, and the beauty was there were also a study from Andrei Shevchenko's group published, and the bioinformaticians developed here a signature which has also a nice ROC curve. And we could use Andrei's data to validate this signature. And this is, of course, only possible if we have quantitative data. And the second nice thing is here this signature has also some prognostic benefit. Yeah, and yeah, this is just a biomarker example. But the main message is if we use here standard and if we have very quantitative values, we can do such nice things. That this brings me to the end, and this is standards in lipidomics, and where we already introduced that. We started that a couple of years ago because we saw really misidentifications and we questioned several studies. Therefore, we founded here the lipidomics standard initiatives. And there were more comments. And the not so good news is it still continues. Yeah. Here, these quality problems, and therefore there was a lot of discussions with meetings. Maybe some of you participated here, and finally we developed here a checklist, which was just recently published here, and this is online available. And I can only advertise, to use it. So this is still beta phase. So if you have some problems with this or questions related to that, please tell us. And what does this questionnaire ask? It asks about identification. How, for instance, it asks how did you quantify your PC? Did you check here for isotopic correction? Or did you check for an isobaric overlap also? These quality-related questions were in that checklist, and you could submit that checklist together with your study data. And this makes it for reviewers quite easy to see how this particular data were generated. And this is a nice way to increase quality. And where we already mentioned, we have the nice situation in the lipidomics field that we have really a nomenclature which is useful to describe the mass spec data, different hierarchies. I think I don't have to tell you, and Mari mentioned already, this is hosted here in the lipid maps site. And if there are questions, I think you please submit there. And this is subject to the development and updates, of course, regularly. And with that, I'm at the end, and want to really thank all these people here, and our group, Marcus in particular, Josef and Klaus-Peter, Christa, Kim, and also the main people here for the checklist, but there were many more involved in that. And here Fritz, Valerie mentioned it already with the nomenclature. And want to thank you for your attention, and I'm happy to take your questions.
Thank you, Gerhard. That was great. I guess one thing just to mention at the end was when you talked about the shorthand being on lipid maps, we did today launch a community board which allows people to interact with lipid maps, ask questions, suggest new features, whatever. And I think Matt has already set up one thread on shorthand, So people can go in there already and ask questions, and so yeah, absolutely. So we have a few questions. So the first one is Akash Bhaskar. You are allowed to unmute yourself if you'd like to go ahead. Akash, unmute. Okay, well,
Hello?
Yeah, there you go. Yeah, we can hear
Hello.
you. Yeah.
Hello, sir. I'm Akash Bhaskar, a PhD student from India. So
Hello.
just wanted to know, so there are multiple columns which can separate both polar and non-polar lipid species. So if you are using those columns, for example, amide helic column, so how reliable are those columns to separate both the lipid types in single run?
I think, this is, you refer to the helic methods. I cannot comment on the particular brand of columns, but this works. Yeah, you get a nice lipid class separations. I think you need to have some practice, to run these helic columns properly. But I think there you can get really advice also from the manufacturers. And at the end, you get here robust and reproducible retention times also in a batch. But clearly, it's not like a reverse phase method. Yeah, takes more equilibration. Yeah, but at the end, it's fine. And I think if you also check Michael Hoczabek's data, this works. And there are several publications. Yeah, you may check the different columns are tested. But yeah, I really can recommend, and you get this polar selectivity. Yeah, does it answer your question?
Yes.
Okay, thank you. So next question, Dorota Skoronska. You should be able to speak now. Dorota, are you there?
Oh, yes.
Yeah,
yes. Only now I can see. Hi. It was fantastic talk. Thank you so much. I mean, so many tips, and I definitely will go through the literature that you suggested. I have a particular question. How would you suggest to extract lipids from Sepharose beads or from some, potential beads that could dissolve in chloroform?
Yeah, that would have been my question. I have no experience, I have to say, with the beads. So you think Sepharose dissolves in chloroform? It's sugar-based material, isn't it?
Yes, but for some reason we are getting not very good results.
Okay.
So we have extracted some subdomains, and I'm very, we want to extract everything. So re-extraction with methanol method, or I mean, like,
did you use here really these chloroform-based methods or MTBE, or which kind of extraction did you use?
So we tried the Blyer.
Okay.
Yeah, exactly. This method we tried, and it is definitely damaging the beads.
It's damaging, and you get them dissolved in the chloroform phase at the end, or?
So yes, it actually makes. We didn't dare to put it on a mass spec because it doesn't look very.
I don't blame you.
Yeah, I don't know. Usually, our experience is if you do really a blind tire, you have usually a quite clean chloroform phase, and there shouldn't be much sugar in that phase, because it's too polar to enter the chloroform phase. But I'm not sure why it dissolves in chloroform. This is strange for me, because they have also methanol, but no good idea and no experience. Sorry.
Okay. Well, thank you.
Yeah,
if you don't know, who knows, right?
Exactly. Yeah. Georgius Isaac, go ahead.
Yep. Hello, can you hear me?
Yeah.
So you showed, thank you for the talk, it was great. You showed different extraction methods and extraction solvent. So in the conclusion, do you have any recommended extraction solvent for both coverage and recovery?
So as you realize from the talk, we are great fans of chloroform-based methods. Yeah. So Bleianteil, feucht extractions. This is what most people in the lipidomics use. MTBE, is a new in terms of toxicity, has advantages, and also you have the can recover the upper fractions, of course. So this would be my preferred methods. So the one-phase extractions, I think you need really to evaluate and use a more non-polar solvent, I think. Yeah, these butanol, methanol mixtures, and with a higher fraction. I think Peter Michael is using that 3:1, and with a high solvent to sample ratio. Then it may be fine. But yeah, I would use the two-phase extractions, because then you have the nice situation that you remove really these non-polar contaminants. Yeah, for the flow injection methods, I would never use a one-phase. We also tried it, and you see, especially in the in that study, we were asked to really infuse also this one-phase extracts. And in positive ion mode, we were astonished. It looks more or less similar, but in negative ion mode, you see a lot of additional peaks, and you see that there's a lot of things present in this extract. So more the cleaner, the better, I would say. And you get cleaner extracts with a two-phase.
Yeah. Okay. Thanks.
Welcome.
Okay, Christina Ferreira.
Hi, can you hear me?
Mm-hmm.
Hi.
Yeah.
Good to hear.
Hi, Gerhard. So I have been doing the same as Valerie over the years. I have been looking for Gerhard's comments every time and advice every time I have questions. So I'll keep doing it. And thank you so much, Gerhard, for this talk. I know, so the value of quantification for the analysis is very evident, but I would like to hear what are your thoughts about lipid exploratory or first pass analysis using just lipid profiling without internal standards. So this data provides, is an indirect picture of the composition of the tissue, and for example, membrane fluidity. This is relevant, and this type of data is being used for intraoperative diagnostics using DESI and using the mass spec pen, and it shows very nice correlation with tumor and cell concentration, even with some mutations in this cancer. So I'd just like to hear your comments about, no quantitative analysis.
Yeah. I think these are very nice applications. Yeah, I know this mass spec pen and all that, very, very good. But this is more pattern recognition. So I'm not sure how quantitative these data are, these spectra at the end. Yeah, and you see differences clearly. But I'm not sure, did someone study? I'm not so, we have no experience in that field. Did someone study how quantitative these DESI methods are? Yeah, I'm not sure. Yeah, we know it from the mass spec imaging things. These are critical. Yeah. So there it's not easy. A lot of artifacts you see in source fragmentation if you think of multi-imaging. But this is a different story than DESI, of course. I'm not sure. I have no experience, but great story to really differentiate these different tissues, and also to see really healthy and pathologic tissue. This obviously works, and I clearly agree. You don't need a quantitation for that. Yeah, you just need a peak pattern. Yeah, and so there, but the next step to have this for biology, I would really prefer to have like a laser dissection fraction of that tissue, and really to do a quantitative approach. Yeah.
Okay, thank you.
Thank you.
Okay, so Megan Jankowski, you have a question which I actually think Gerhard would answer, but it may also need to go to Matt Conroy because it relates to Lipid Maps website. I don't know if you can read it already there. So I'm going to unmute Matt as well, and then you can take the question first, Gerhard, and we'll see then if Matt needs to reply. So go ahead there, Megan.
Hi there. Can you hear me?
Hi. Yeah.
Yeah.
Thank you for your talk today. I'm certainly going to check out that checklist. But more immediately, I do have a data set that is from a fungus, and I'm dealing with a lot of issues where I have this great lipid maps, identified lipids, and that nomenclature, but getting that to match up with any database to understand what biological pathways might be involved is proving very difficult. So any insights on certain databases or programs like LipoStar, I'm willing to check out anything and everything, or maybe Lipid Maps has the answer and I didn't realize it.
Because you're specifically dealing with fungal lipids of what type?
We also do mammalian things in our lab, but I'm having more trouble with fungi.
Yeah, that doesn't surprise me. Gerhard, you go first.
No clue, honestly, and I think you have more experts, Valerie and Matt also.
Matt,
so this
over to you.
Yeah.
Matt, you're there. We can't hear you. He has unmuted himself, but we can't hear him. Oh, is his microphone not working? Give him a second. I think his microphone must be working. Well, while he tries to work that out, I think the answer to that is that the lipid map structure database was originally set up mainly for mammalian. So you should get pretty good matches there that you know that match most of what anything that doesn't match may well be it's not going to it could be something unknown or it could be something else. But most of the known ones should all be there. But when it comes to non-mammalian lipid structures, we've started bringing them in over the last few years. We brought in a plant database, and really, you know, there have been many other lipids come in from other sources, and at the moment we're really relying on the community to flag them to us so that we can increase the representation of non-mammalian lipids in LMSD to really help address the problem that you raised, Megan. So I suspect the answer is that we don't have that many fungal lipids in there. Matt, you're still unable to speak, aren't you? It looks like. What a shame. But anyway, can I suggest, Megan, that if you can get in touch with Matt through the new community board that's just been set up on Lipid Maps. You can start a thread on fungal lipids and get people talking about this, and where people have fungal lipids that are not represented, they can send in the structures and they can then be put into LMSD so that we have them there.
Okay. Any other tools for the more general issue of just getting lipid maps to be converted to KEG IDs or TEBI, anything? It seems like there's a lot of missing connections between lipid maps level of knowledge and some of these other database IDs.
Yeah, I mean, there's a lot there. I mean, KEBI IDs are represented there where they align with lipid maps IDs. KEG, I'm not so sure. Again, it's a shame that Matt's not there, but we have got links to many other related databases through individual entries. I guess it depends on what kind of search you're trying to do, whether or not we can help you do that. So the best thing is to get in touch, I think, with Matt through the community board, and we'll see how we can help answer the specific of what it is you're trying to do exactly.
Okay, well, thank you very much.
Thanks, Megan. So last, I know we've gone over time a little bit, but he says he's unmuted, but his microphone isn't working. We might have some fungal lipids, but in terms of pathways, no data yet. So if you have some, Megan, send it in and we can get it put in. So a question, we are a bit over time, but let's go on. We've only got another couple. So Roland, I'm going to get you to unmute. Go ahead. You should be able to speak. Roland? Don't tell me your microphone's not working either. I think we have another one. Roland, are you there? Okay.
Yeah, but I can read the question, or?
Well, we have another one, and I prefer to see if we can help them speak. So let's see if he can work it out. But we'll go to Inchi Barut. Are you able to speak there?
Hello.
Hello.
Thank you for your great talk. I have a question about the incubation time of stable isotopes in cells. Is there a converging time that you try, like time course, and how long generally does it take on average for an efficient tagging?
I think that depends very much on the cell type, and the condition you are using. So if you have quite active metabolism, as you see it here in vivo for the liver, you see after a few minutes you see already nice peaks, and after hours you get really nice spectra also for plasma, for instance. In the cells, typically we have done studies in monocytes, macrophages. There we use as the first time point about 15 to 30 minutes, then 2 hours, 5 hours, 4 or 5 hours. This is usually the time frame we are using, but yeah, it depends really on the type of cells you are using, I would say, and which isotopes you are feeding. Whether you use this head group labels, or you can also use fatty acids, of course, to label, or acetate to label the fatty acid synthesis. There are many ways to label lipids, and this is a particular question. What is your question, and what do you want to see?
Okay, so Matt says to Megan, KEGG IDs are there where they match. Unichem is a useful tool. So hopefully that helps, but Megan, do get in touch. And then the last question from Roland, his microphone is not working. So Gerhard, if you want to go and read that out and ask.
Yeah, it looks like it's not working.
No, it's working.
No, it's working. Oh, great.
Go for it.
Yeah, great overview, Gerhard. Thank you very much.
Thank you.
So my question was really in the one-phase extraction efficiency data, which was really also nicely presented, which is really nice view to see it. So on the solvent dependency, do you have any data on the effect of chain lengths within a certain lipid class, especially the non-polar lipid classes, on the solvent extraction efficiency?
Yeah, we have that data. This is in the manuscript, in the supplementary data. You find all species data also. And I have to say, it's weird for the non-polar, because if you do not extract anything almost, you see only the major ones and completely weird profiles. I think interesting if you go to the more polar, for instance, a PC, then you see improved recovery or higher recovery for the more polar ones. If you have a proper sufficient extraction, you see no change in the profiles. Same for if you're isopropanol or something. But if you go to the acetonitrile or methanol, you see higher recovery for this PC3064, for instance, and this is because this has a better solubility. The PUFAs have higher solubility in these polar ones, so you see some trend. If you have improper extraction.
Okay, thank you.
Okay.
You're welcome.
Okay, thank you. Well, I think we've exhausted all the questions and we've gone over time. So it just remains for me to say thank you very much, Gerhard. That was great, and it will be up on the Lipid Maps YouTube channel in the next day or two. So thank you, Gerhard, again, and look forward to seeing everybody next month for our next webinar.
Thank you for the chance to present here.
Bye.
Bye-bye.